I want to bring up to the stage Tom Lipkin, my boss and colleague who represents the Innovation Fund, to talk about our efforts in commercialization and executing on that. Dr. Tom Lipkin.
Thank you, Matthew. It's thrilling to be here. My name is Tom Lipkin. I am the managing director of the UCLA Innovation Fund. We are an internal proof of concept fund here at UCLA. And I'm excited to be hosting the Crossing the Chasm pitch showcase event. So first, a few words on innovation entrepreneurship here at UCLA. UCLA is an incredibly entrepreneurial campus. We have a rich, robust ecosystem. I work at TDG, the Tech Transfer Office. We protect the intellectual property across campus. And we receive over 300 new technologies that come into our office each year. We also spin off, on average, 20 startup companies per year. These are external companies that have licensed intellectual property from UCLA, and they really do run the gamut. You see a few of our startup companies in the medtech space in the lower right corner of the slide, and there's several others that I just couldn't fit here as well. And you ask yourself why, how does UCLA, why are they different, and how do they have such a robust entrepreneurial ecosystem? A lot of that comes down to our faculty members, right? I mentioned the high number of disclosure rates for those faculty members in the audience. We encourage those disclosures, keep them coming. But also we benefit from our geography. We have a very dense campus, right? Population's quite high, and you have these collision events here at UCLA that you might not have at other peer institutions, right? You have clinicians with engineers, with scientists, and because of that, our technologies are that much more fertile and more advanced and ripe for commercialization. No talk on entrepreneurship is complete without an obligatory Valley of Death slide, so here it is. This is the Valley of Death, the Chasm of Chaos, whatever you want to call it. But basically, there is this rift that exists between where academic science and funding stops and where outside parties are going to fund and advance this technology. And so with this challenging funding environment right now, it's impingent upon outside investors to take early stage risks and fund these promising projects. But also here within the university, we need to do more to provide support for our entrepreneurial faculty, like those you're going to hear of later this session, on how to advance and move their projects further towards that path of commercialization. I just want to highlight a few of the resources we have here. In terms of our pipeline, I mentioned our rich research that happens here at UCLA, but we also have two programs that I want to highlight really quickly. The biodesign program. This is a program that is run by Dr. Jennifer McKaney. This is a collaboration with UCLA Health, DGSOM, and Anderson, our business school. And it does needs-based analysis. So it goes into the clinic and talks to physicians on unmet medical needs, and it works with them to develop medical devices or ideate around medical devices that could help solve these crucial problems. It's a year-long fellowship. There's a table outside. I encourage you to go stop by and get more information on this program. We also benefit from being an NSF I-Core site and hub. So this is a program run by Dr. Mel Ulu-Chakriolu. She unfortunately couldn't be here today, but this is a program where if you're an entrepreneurial faculty member, you can apply to this program. It's run through the School of Engineering, and they will arm you and equip you with the tools to do customer discovery. So these are, it's a didactic plan where you develop your questionnaires and you go to Voice of Customer, and that will help influence your ultimate business plan. There's also funding available, so I encourage those to take advantage of that. And one of the teams that you're going to hear of later today actually benefited from that program, and maybe they can speak to that or answer any questions that you might have. And then finally, our program, the Innovation Fund. So this is an internal proof of concept fund, non-dilutive capital. And again, we're trying to bridge that valley of death the best we can by sourcing, funding, advancing these promising projects as well. For those of you that are in need of additional funding, just really quickly, we provide up to $150,000 in funding. We bring an outside advisory panel of investors and industry folks to give the necessary feedback and to move these projects to hit key inflection points, to de-risk them, to make them more attractive to outside parties. And for those of you in the audience that are UCLA faculty members, good news, our application cycle is open. It closes very soon. It was going to be this Friday, but we've extended it. So you have over the weekend, but I encourage you to apply if you are interested in advancing a medtech project or a therapeutics project. It is due Monday, May 26th, 11:59 p.m. So please encourage you to apply. Here's a QR code. You can find myself, You can find Matthew afterwards, and we're happy to answer any questions that you have. So with that, thank you. I am excited to invite up our esteemed panelists. This is going to be a pitch session. So the teams that you're about to hear from today have received funding from the Innovation Fund, and they're here to tell you about their project. There's going to be an 8-minute pitch followed by a 2-minute Q&A from you, the audience. So I encourage you to get your questions ready. And then I'm going to invite all of the panelists up at the very end, and we'll take a group questions. So up first is Dr. Ann Andrews and Marlee Goldstein. We're talking about their project Femsync.
Hi, everyone. We are Femsync, and we are developing the first non-invasive wearable for continuous female hormone monitoring. This past winter, I was hospitalized for blood clots in my lungs. And because hormonal birth control is a contributing risk factor, I was told I would have to go off the pill indefinitely. For the first time in almost 15 years, I started to experience the hormonal fluctuations that accompany a menstrual cycle, and they hit me like a truck. I immediately noticed changes in my cognition, energy, sleep, and mood on a daily basis. So I researched online, I purchased wearables, and I downloaded cycle tracking apps to try to understand how my symptoms were matched up to these hormonal changes and if what I was experiencing was normal. But I experienced one huge problem. There is currently nothing on the market that can measure my actual hormone levels continuously. This is what women's 3 most important reproductive hormones, estrogen, progesterone, and luteinizing hormone, should look like over the course of a 28-day cycle. However, in practice, many women do not experience a normal period month over month, and we can see changes based on exercise, travel, stress, nutrition, alcohol, medication change, you name it. And without actual information of what's going on in our individual bodies, we're left confused and spending hours convincing doctors to take our menopausal, fertility, and menstrual symptoms seriously. But what if I knew exactly what was happening in my individual body? I could understand how my symptoms match up to my hormonal changes, and I could plan my exercise, sleep, nutrition, and key events in my life around those phases to maximize my productivity and energy. She could identify potential hormonal irregularities up to 7 years before current medical standard, enabling her to have early family planning conversations with her doctor and increasing her chances of having a family. She could know for a fact that she was entering perimenopause, allowing her to better measure her symptoms with her doctor and keep her doing the activities she loves. And she could avoid unwanted pregnancy without hormonal contraceptives. So what if we knew? Women's hormones affect our bodies on a daily basis and throughout every phase of our lives. 50% of women have a problematic cycle, 15% of women struggle to get or stay pregnant, and 80% of perimenopausal women face symptoms that negatively impede their daily lives. Yet for most of us, our hormones are a complete mystery. At Femsync, we're developing the first non-invasive wearable that measures reproductive hormone levels through passive sweat on the surface of the skin. No needles or pee sticks required. In practice, this looks like a patch that's a sticker that is disposable that you can place anywhere on your body for up to 30 days. Once in the morning and once in the evening, it will automatically collect sweat from your skin and measure the concentrations of those 3 key hormones I mentioned, estrogen, progesterone, and luteinizing hormone. This data is then used with our user-facing app and our algorithm to provide women with insights and actionable trends so that they can better improve their health. For the first time, women will be able to track their changes in their hormones over months, years, and eventually decades.
So I'd like to tell you a little bit more about our sensing technology. We've carried out research for years, and it's enabled us to break through a fundamental problem in biosensing. Namely, we've developed a biosensor that enables us to measure hormones at the ultra-low levels at which they occur in sweat and other biofluids. Our sensors are modular, they're generalizable, they're manufacturable, and they do not require any added reagents or sample processing. First, the sensors actually use a small piece of DNA, and that DNA can selectively recognize each of the 3 reproductive hormones. Once hormone binding occurs, it's transduced into a transistor-related current, which is intrinsically amplified. We have developed a wearable prototype, and that prototype, which you see on the right, includes sweat sampling, electronics, and a readout. We have 4 patents filed by UCLA, both at the U.S. level and worldwide, that protects our general sensor strategy and our specific applications in monitoring female hormones, as well as the major stress hormone cortisol. Currently, there are no other products on the market like ours. We will be the first product that will enable wearable, continuous, non-invasive monitoring of estradiol, progesterone, and luteinizing hormone in a cost-effective manner. About a third of women already report using cycle tracking apps like Flow to try to monitor their cycles. However, these apps require user-inputted data, and they're unable to deal with cycle irregularities. In fact, about half of women never experience a normal period. And most women, as Marley said, have impacts over time that give them cycle irregularities associated with stress, sleep disturbances, intense exercise, or illness. And so these types of apps cannot provide the accuracy that women need. We have commercial wearables like Aura and Whoop that are based on body temperature measurements. And while more accurate, these devices still fail to give women the quantitative hormone data that they need to ultimately understand themselves and with their doctors what drives their cycles and what drives fertility. Finally, we have blood tests and urine, which are more accurate but are much more costly. In addition, they require intense user input. Nobody really wants the discomfort and the inconvenience of repeated blood draws, especially for months or maybe years. And most women tell us they literally do not want to get up in the morning and have to remember to pee in a cup. We have done market research that involves surveys and personal interviews with more than 600 women to find out what women need and want. And this has helped us to define our go-to-market strategy, which includes going to market in the health and wellness sector. This will enable us to reach the largest numbers of women and to work with women in terms of gaining their hormonal data across their reproductive lifespans. This will enable also the largest customer lifetime value and the lowest regulatory barriers with the FDA and the largest market size.
Speaking of market size, this is a huge opportunity as women are 50% of the population. Looking at the total addressable market, we see a potential $45 billion opportunity. And these are markets that are growing rapidly, with the fertility and hormone wearable segment alone growing at 36% CAGR. Even though we're going to market in the direct-to-consumer wellness segment, we see partnerships as really the key to growth and scaling. We are looking at primarily 4 types of partnerships, including health wearables like Aura or Whoop, cycle tracking apps like Flow or Natural Cycles, nutrition and exercise apps such as Noom or Strava, and fertility and women's health care such as Kindbody or Maven. And with these listed on the screen alone, these icons, it's almost 500 million additional potential users.
So we have deep expertise in our team. I am CEO and Chief Science Officer. I'm a professor at UCLA with 30 years experience in biochemistry, bioengineering, and mental health research. My co-founder and partner, Marley Goldstein, has 9 years experience in health technologies at Deloitte and with growth strategy at Uber. Our lead chemist is Dr. Noelle Mitchell, and we're advised currently by Josh Sackman, who is president and founder of Applied VR and also a Techstars mentor. We're currently, we've raised about $10 million in basic research funding that's gotten us to this point. We have about 6 months runway in our pre-seed funding that's going to enable us to do research, to do multiplex monitoring of hormones, increase the stability and lifetime of our sensors, and begin to do monitoring in real human sweat. We're looking to raise about $500,000 to enable us to complete these de-risking experiments and to carry out limited studies in cycling women and to benchmark against gold standards. And so finally, I'd like to thank you very much for listening to our vision of the future for women's health and hormone monitoring, and we are looking forward to your questions. Thank you.
Thank you. Anyway, in my experience, there's maybe 5 pillars that you just have to go through. There are no shortcuts, unfortunately. And I love this idea in technology. The one that sticks out to me is the publications or the clinical trials of data, proving the how and the why and what difference will this make. Can you walk us through that clinical trial plan and where it starts and kind of what those ideas will lead to? I mean, what type of trials or what type of data, and if you collect it, will that drive a reimbursement outcome, for example,
Right.
or a user outcome that of some interest? Thanks.
Thanks for your question. I'm going to try to answer that quickly, and we can talk more about that. Clinical trials, other than showing that our sensors measure what we say we're measuring, are the only thing that we're really needed to get into that health and wellness market with the FDA. If I understand you correctly, you're really talking about more medically oriented clinical trials. That's part of our longer term strategy. There really are no data like this that exists. So we're really in a very new space. We have no way to kind of collect this level of data density on women. So we'll be working with the FDA to design these trials. We have a number of different patient populations that we're interested in looking at. As Marley said, for instance, many women who suffer from PCOS, time to diagnosis is 7 years. And these kinds of changes in hormone levels at the individualized and personal level can probably be picked up by our sensors. So those are the kinds of things we'll be looking for in clinical trials. And we will be looking for payer reimbursement to drive, decision-making for doctors, enable women to take hormone data directly to their doctors with their concerns. Yeah. Thanks. Yes.
I'm wondering how you thought about what sort of information you'll give back to people in terms of what they should do with the data. How do they interpret the data that they're getting back from their sensors? For instance, if somebody gets pregnant when they're told that they're in a part of a cycle where they shouldn't, what's the liability risk to the company? And, questions like that. How have you thought about how you're going to give data back to people?
I can take some of the kind of data and insights questions here. There's a couple things we've considered and that we've gotten from those 600 user interviews and survey responses, which is exactly this, that women want actionable data. So in some senses, we are going to have to combine our insights with additional research as we're collecting this data from women, because as we've mentioned, there's currently no continuous monitoring of hormones happening. So one of the reasons we want to get into the wellness segment is to understand exactly with growing this flywheel of data, what are the trends that we see in women at different phases of their cycle. And one thing we got from our user interviews too is the ability to kind of like do experiments within the application. So if they're trying a different type of nutrition regimen, trying a different type of medication, trying an intervention that they've planned with their doctor, giving them insights on how that has changed their hormone levels or symptoms over time. And that will be really key to helping to understand kind of the actionable side of it. Hopefully that answers your question, because we definitely agree that just hard data is not always more data is not always better. We want to give women really personalized insights into A, their patterns, and B, what they can do about it.
I think we'll have to take the rest of the questions later, if I understand. Yeah. Thanks for having me.
Thank you, everyone.
Thanks.
Excellent. I'd like to invite our next speaker, Dr. Igor Barjaktarevic. He is speaking about his project, the System for Asymmetric Flow Regulation, or SAFR.
Thank you, Tom, for the introduction, and thanks for the organizers for having me here and giving me an opportunity to introduce the concept of SAFR, which is, I think that this is where it starts, which is System for Asymmetric Flow Regulation, the first of its kind device that aims to really provide the customized ventilation to each of the 2 lungs in mechanically ventilated patients. So coming from an intensivist, I can tell you that mechanical ventilators save lives for over 8 decades. And besides, obviously very high number of patients that we actually have, the number is increasing year by year. The indication to put a patient in mechanical ventilation in intensive care unit is associated with high mortality, and let's to say with high healthcare resources utilization and costs. And interestingly, the small number of patients that require the extended duration of mechanical ventilation actually consumes the most of healthcare resource utilization. Now, while mechanical ventilators without doubt save lives, they also hurt. Notorious ventilator-induced lung injury happens, and what actually it does is that based on the barotrauma, based on the concept in which normally we breathe with the negative pressure ventilation, our muscles take the negative, make a subatmospheric pressure inside, the air gets sucked in. In contrast, with the positive pressure ventilators, the pressures are pushing the air in. Pushing the air in actually leads to something that actually leads to an injury that I'm sorry that this is actually old version of presentation that I don't have everything that I wanted to say. But actually the lungs under the pressure gets injured, gets rugged, swollen, and eventually like useless. Now there is one more caveat to this. The pathologic changes that lead to the need for mechanical ventilation are not the same. Some are just inherently asymmetric, such as lobar pneumonia or effusions or pneumothoraces. But some even more uniform, such as acute respiratory distress syndrome, actually, as we showed in the studies that we published, really have almost 1 out of 5 patients with severely asymmetric injury. And we showed that actually these patients have longer duration of mechanical ventilation and poorer outcomes. Now, what is the problem? The problem is that despite us having 2 lungs, 6 lobes, 18 segments, millions of alveoli, the ventilator sees the lung as single system. And when this actually happens, what we have is actually, as you can see in that video, is that if normal lung would be looking, if normal lung kind of equally gets distribution of gases, once you have the injury on one side, what happens is that not only that area of the lung gets underventilated and on insufficient gas, it's also that the other healthier portions of lung get much more pressure, more air than what they needed, get hyperventilated, and eventually get injured. The study of UK group that really looked at the people who received single lung transplant, so they keep one of the old lungs, hyperinflated, emphysematous, hypercompliant lung, and a new normal lung, they showed that actually those with the biggest difference in compliance of the 2 lungs actually have the poorer outcomes. I could not resist but not to show you this picture, and this picture actually really comes from almost led to and inspired this project. The midst of pandemic, COVID pandemic 2020, we're in the ICU with people who do have like really trashed lungs that are severe lung injury. We're trying to help it with paralyzing, proning. We're doing like maximal oxygen, high pressures. And one paradox that we notice and phenomenon is that in this highly pressurized system, if we put additional focal pressure in certain areas, which by the way manifests here with the sandbags and some tools and the belt to really tighten this up. And I think the cup here is just artistic detail that I had to put just to show the paradox of the situation. We are actually getting that the pressure in the system is lower and the oxygenation is better. And that's what inspired us to actually get to develop the system for asymmetric flow regulation, which really aims to provide personalized, customized ventilation to 2 lungs independently. It obviously tackles the concept of trying to tackle the problem of ventilator-associated injury. And it's simple but revolutionary concept that really focuses on compartmentalized lung ventilation, and yet it incorporates in current standards of mechanical ventilation and circuits. It's proven in the animal models and the bench models, as I'm going to show in a second. And obviously impact can reach pretty far, both in terms of improving patient outcomes as well as healthcare costs. So what do we have? This is just the scheme that in mechanical ventilation, we have the ventilator in circuit on one hand side, we have the patient on the other side. The ventilator is connected to the patient with endotracheal tube. In this particular concept, we actually are using the Solve tube. Double lumen tubes are used in OR and the surgical ORs for decades. What we actually got, like, and we're very happy that at UCLA near Hoffman developed the smaller, easier to use universal tube that is easy to use in medical ICU. So we use this double lumen tube. And then we have the device that really pretty much looks like a piece that is incorporated between the ventilator circuit and the double lumen tube. What is it composed of is the flow regulator, which pretty much has connects to each of the tube lumens. And it has ability to control the inside lumen of that portion of the regulator. Thus, by engaging and decreasing the lumen of one side, shifts the flow to another side. Now, to exactly know what are we achieving with this, we have the pressure and sensor regulators, which really tell us how much air is going through each of the limbs and really reflect the mechanics of the lungs, both static and dynamic, as well as end-tidal CO2. So end-tidal monitors of carbon dioxide, which reflect the physiology and the ventilation in each of the lungs and the consequence of the distribution of the air. Now, that is the device that really connects to the circuit. The device connects to a housing that has a display with a continuous monitoring system of flow, pressure, and end-tidal CO2 of each lung. Now, UCLA helped us patent the product in the United States and Europe. We actually received the award of TDG. We shifted the award and all the finances to industrial development, who got the product to look something like this on my desk in the office right now. And we tested that prototype on the bench models. And on bench models, we used like sophisticated simulators, lung simulators, that actually are able to capture, to give the compliance, resistance, volumes. And we use 2 models, left and right lung. And using our device, we really actually try to really show first, put different resistances of the airways in one and another lung. So make one 10 times higher resistance. Normally, as expected, the lung that has high resistance will receive less air, as you can see in the first and right. But then we are able, using the SAFR, we're able to neutralize this effect, to equalize the pressure, and even reverse. Similarly, when we change compliance of the lungs, when we make one lung stiffer than the other, stiffer lung will receive less air first. We're able to equalize. We're able to even reverse the concept completely. More recently, we completed the pig animal model tests. So pig intubated with the double lumen tube using the SAFR. We were able to show that we can really distribute the air to left and right lung as we wish and get the feedback and know exactly what are the pressures, flows, and end-tidal CO2s. We were able to actually, in the open lung model, when we compress one lung and make it stiffer, we're able to show that we can neutralize this effect and reverse the airflow distribution. And most importantly, we also, as what we could not do on the bench model, we showed that physiology works, that end-tidal CO2, that we can see from each lung what's happening and what is the impact of the changes that we make. So with us kind of anticipating what are anticipated cost savings, I think that first focus can be really on the patient outcomes. I think that reducing the cost, the day of mechanical ventilation is already by 1 day reduces by $15,000. So what are our next steps? So obviously, I'm a clinician. I try to generate more data. We have publications that are already published, some in preparations. We are at the same time really trying to partner with and develop kind of the network so that we actually collect more funds and able to get to the next stage. Obviously, I would like to just thank, besides you, TDG, thanks my partner, Glenn Merowitz, the engineer from SpaceX, who joined BioDesign, figure out what we're doing and help me get where we are. Of course, all the other people listed here. Thank you very much. I hope that I did not break the time.
Unfortunately, we're running a little behind schedule, so we're going to have to forego questions right now. So apologies.
Thank you.
So our last speaker is Dr. Liang Gao and his project, Lift Photonics.
I'm a social professor of bioengineering here at UCLA. I'm also a co-founder of the Lift Photonics. So in this talk, I want to talk about our recent effort in developing this new technology called real-time three-dimensional film and how this technology can potentially revolutionize these surgical operations. In the realm of the cancer treatment, although there are many options exist, the surgery still remain the standard of care for patients, especially for the late stages. However, in the OR room, the surgeon frequently facing this kind of challenge that tumors are very hard to distinguish from these normal tissues. This leading to the cases where tumors cannot be totally resected and require this further surgical resection. This leads to this can create these situations of these significant social economic burdens, not only for the patients, but also for the hospitals and insurance companies. So here, to address this challenge, we aim to developing this label-free tumor visualization method utilizing fluorescence lifetime imaging. And our technology can help the surgeons differentiate tumors from normal tissues with high accuracy. So to reduce these surgical errors as well as these needs for the reoperation. Fluorescent lifetime imaging has been widely used in the biomedical research. Upon excitation, most of the molecules can generate fluorescence, like the glowing stick. But the fluorescence emission will not take forever. If you have finite duration in time, this duration is a signature for the molecules. By detecting this fluorescent lifetime, we can differentiate different molecules and further derive the tissue composition. This information can be used to facilitate the disease diagnosis. Among various applications, FLIM has been demonstrated most effective in the label-free assessment of tumor metabolism. This is because for the tumors, they grow fast, so they consume more energy. Their metabolic rate is much higher than normal tissues. By detecting these elevated metabolisms using autofluorescence lifetime, we can differentiate these tumors from the normal cells, and further using this information to guide the surgery. And due to this reason, FLIM has been widely considered as a very promising tool for the intraoperative surgical guidance. However, FLIM suffers from two major drawbacks. First of all, it's very slow. And second, the current FLIM cameras can only capture two-dimensional images. This limits their application in the surgical scenario. Our technology, what we call light-field tomographic FLIM cameras, which for the first time can enable three-dimensional fluorescent lifetime imaging in real time. Our technology will revolutionize the intraoperative cancer imaging by simultaneously providing the 3D visualizations of tumors in the entire surgical resection cavity, as well as providing the label-free cancer imaging. As I mentioned earlier, FLIM is great, but it's slow. The reason is that to capture the two-dimensional or three-dimensional images, the conventional FLIM cameras have to rely on scanning. For example, for the confocal FLIM, it has using this tiny focus spot and scan it across the entire field of view. The reliance on scanning slows down the image acquisitions, and it can introduce significant problems when we image something dynamic scenes. For example, the motion of patients can introduce significant motion artifacts. And due to these lengthy acquisitions, most of the current FLIM systems, they are limited to capture two-dimensional images. This restricts their application in the surgical scenario because these 3D visualizations of the surgical resection cavities is necessary. Our technology targets the physicians by providing this 3D visualization of tumors. This information can be used to guide the surgical resection with much enhanced accuracy, while minimizing the post-surgery cancer recurrence. The lift film technology is based on patent technology developed in my lab here at UCLA. Our design is actually inspired by nature. Instead of capturing the image using a single lens, like the camera lens, we use an array of lenslets. This design mimics the compound eyes in the insects, like the flies. Each lenslet on this array captures a unique view or perspective image of the three-dimensional object. By deriving this disparate information from these adjacent views, we can construct the three-dimensional image in a single snapshot. Using the lift film, we can record this 3D film imaging in real time. With the help of the TDG Innovation Funds, last year we developed this compact module design for our device. A left side picture showing this our device it can easily fit it into this carry-on luggage. And the right side picture showing when we connect our device to the commercial microscope, providing these additional 3D visualizations of fluorescence lifetime information. Using our device, we first demonstrated in the cell imaging. Here we image a mouse kidney sections stained with two fluorescent dyes. Using our device, we can capture this three-dimensional image in a single snapshot with a high spatial resolution, as well as providing the fluorescent lifetime information in each pixel in the 3D image. And by using these lifetime signatures, we can differentiate these fluorescent dye together and mix them into different channels and rendering this three-dimensional visualization image. We further test this technology in the cancer imaging. Working with our clinical collaborators, we imaged these biopsy samples from lung cancer patients. By imaging a large field of view, we found a significant heterogeneity in fluorescent lifetime. And this information, heterogeneity, is correlated well with HE stained ground truth pathological results. By further applying the phasor analysis, we can separate these normal tissues from these cancerous tissues providing guidance for the tumor margin assessment. Moreover, we test this technology in the brain tumor as well. Here, the middle column image showing this conventional autofluorescent image captured by the commercial surgical cameras. You can see, using this intensity image, it's very hard to tell which part is tumor and which part is normal tissues. And using our technology, by detecting these elevated metabolisms, we can successfully separate these tumor areas from these normal tissue areas. Besides of cancer imaging, we found our technology can also be broadly applied to other biomedical imaging problems as well. So one of the spotlight application is high-throughput label-free drug screening. Working with our collaborator, we use this film microscope to image this multiple well plate. In each well, we cultured these IPF organelles for this chronic lung disease. By measuring these metabolisms, we can test the drug response of the individual organelles in this multi-well plate without using fluorescent labels. So this approach provides a cost-effective method for high-throughput drug screening for the personalized medicine. Regarding the market, currently we are focusing intraoperative cancer imaging. The total addressable market is expected to grow to $4.4 billion by the year 2030. As within this total market, the service addressable market is film assisted surgery. And this market is expected to grow to $250 million by the year 2030s. Within all the market, the most significant market portion is here in North America, but we also see a rapid growth in Europe and Asia as well. There are several competitive technologies currently also providing this lifetime imaging capability. But you can see here, all our competitive technologies, they rely on scanning. So our technology, this lift film, is the only product on the market can provide this real-time feedback and 3D visualizations for the lifetime information. Dr. Zhongshan Park and I are co-founders of this company. Both of us have over a decade's experience of developing advanced medical imaging devices. We also have several clinical collaborators testing our device in various cancer imaging applications. Currently, we are raising the seed funding to further promote our technology for the clinical trials. We are also seeking the strategic partners, licensing, and investigators. At last, I want to say thank you very much for your patience, and if you have any questions, please send me emails, and we can start from there. Thank you.